Studying.
Okay, so welcome back. This is primary structure of a protein. So we're done with DNA, mRNA, RNA, and tRNA. We're on protein now. So there's 4 different types of structure. Can you point down? Can you see my hands and stuff?
Yes.
Okay. So there's primary, there's secondary, there's tertiary, and there's
Quaternary.
quaternary. Okay. So primary structure is the creation of
I don't know.
peptide bonds between proteins. There's a carboxyl and there's an amine group. A carboxyl is a C double bond O OH, and then the amine group is an NHH. And then there's also an R group, which is the 20 different amino acid R groups, which is adenine, xanthine, thymine, all those things. Anyway, let's forget about those. Those are not important. So let's say this is the carboxyl group and this is the amine group. So the blade is the amine, the back is the carboxyl. Okay, so it'll just go, boop, and then the double bond O will be split between the N and the C and the carbon. And then another one comes on, and then another one, and just build a chain like that all the way across. That's primary structure. Secondary structure is a little different. So secondary structure is what?
I don't know.
Okay, it's the same protein, or the same combination of different proteins, which is now called a protein. We'll just call that the protein. Okay, there's 2 different. There's alpha helix and there's beta folded sheets.
Beta folded cheese?
Folded sheets, which is really, it's beta plated sheets. That's what it's called, plated sheets, which is like folded sheets. Okay, so the alpha helix is, what is a helix? If a DNA is a double helix, what's an alpha helix?
1 helix.
What's a helix?
The shape.
What's the shape of a helix?
5-sided.
No. What's a pentagon?
It's a what?
That's a pentagon.
I don't know what a helix is.
Okay, a helix is a spiral.
Okay.
So it can either, take it and then spiral up by using H bonds, which are
Hydrogen
what? Bonds.
bonds.
Yes.
Okay.
How do hydrogen? No, I'm not going to go to chemistry. Okay, but it can do that by doing hydrogen bonds, or it can fold up and then up like that all the way across. It can fold. It can do both too. It can go from an alpha helix to a folded sheet and then back into a straight line. It depends. Okay, so let's say we did a baited folded sheet. Okay, that's a folded sheet right there. Okay, so now we're done with primary, secondary, and now we're on
Tertiary.
tertiary. What's tertiary structure?
I have no idea.
Tertiary structure is when the protein will fold in on itself to create its 3D structure. So it goes from a line or a helix folded line thingy bending back on itself to make a 3D structure. What creates that 3D structure? What determines the 3D structure? Let's give it a guess. It's a type of bond between what groups?
I have no idea.
R groups.
R
So the
groups.
R groups in the protein will determine its shape, and proteins can be made of. So this is an amino acid. That's what it's, which is the monomer of the protein. Okay, my bad. Okay, so and I don't know what I was going with that. Proteins can be made up of 100 amino acids to 100,000 amino acids, so they can be huge structures. But, imagine that like folding in on itself. Yeah, it's going to be fairly large. Okay, so that's all tertiary structures. There's nothing really complicated about that. It's just folding in on itself because of R groups. Okay, now we're on the last one, which is...
The 4th one.
Which is...
I don't know.
Quaternary.
Quaternary.
Structure. So, we'll go back to this question later, but this is a question for you to think about. What structures are the proteins active at? Yeah. So that's a question just to think about. We'll go back to that. Quaternary it doesn't need to happen. It's to make an enzyme. It's multiple proteins coming together to form an enzyme. A protein after tertiary can already become an enzyme. A protein after secondary can already become an enzyme. Primary, it depends. Not really. But, it can be active after what groups? What stages?
Tertiary.
And?
Secondary.
And?
Primary.
No. It can or it depends on it. It's really complicated at that point. But quaternary too. Quaternary is multiple proteins clumping together to form a larger enzyme.